While they get set up, I'll next introduce Hannah, who's going to be talking to us about statistical methods for single cell proteomics. Hannah is in the pain lab at BYU. She's briefly been a neighbor of ours in the past too. I don't know if virtually or actually here, but she's worked with Professor Vitek as well for an internship. Hannah has explored fragmentation spectra for single cell data. She's also looked at sample sizes for pseudotemporal ordering. So yeah, Hannah, we're looking forward to your talk.
All right, so yeah, my name is Hannah Boekweg. I'm in Sam Payne's lab at BYU, and I've been collaborating with Olga Vitek to come up with statistical methods designed for single cell data that will more accurately detect differentially abundant proteins. So this is the outline of the talk. First, I'm going to go over the problem statement, what people are currently doing, going to present the data sets that we use, then I'll get into the results and the models, and then I'll leave you with our recommendations. So why do we even need a statistical method just for single cell data? So a very common method that is used in bulk proteomics is group comparison, where you average all the proteins, all the healthy subjects, and compare them to the average of all the disease subjects. And you go protein by protein looking for proteins that will be differentially expressed between the two groups. Single cell data gives us an additional layer of replication. So with bulk data, we just had replicates of people, but with single cell data, we have replicates of cell for each person. And with this information comes a lot of questions that we are posed with now. For instance, each cell is like biologically unique. So how do you actually incorporate that cellular heterogeneity into a method that typically just averages all samples together? Also, you have cell type information now. So here I'm showing green cell type and blue cell type. How do you incorporate that into it? Also, you have to have a way of annotating these cell types. Here they're colored, but they don't come that way. We have to do that ourselves. Another question is, what happens if you're not able to sample as many cells from some subjects? Is that going to affect the results? How do we handle that? Can we just ignore it? So these are the kind of questions that we need to answer if we're going to really take advantage of all the information available to us with single cell data. So this is the data matrix kind of showing all the features of single cell data and what we need to incorporate. So we have condition, subject, cell type, and then the cell level information. And we also have the between subject variability, so differences between people, and then the cell variability, which is differences between cell. And this is very important that we incorporate these correctly into the model to improve the model sensitivity and accurately describe the data that we have. So if we're able to do this and incorporate all the features of single cell data, we can ask really interesting questions that bulk experiments are unable to ask. And here are some examples. How are smooth muscle cells changing between healthy patients and patients with a heart aneurysm? How are tumor cells reacting to a small molecule inhibitor compared to somatic cells? And what are differences between T cell subtypes? So this is what people are currently doing. They're currently trying pseudo-replication, where you treat each cell as a biological replicate, and they're trying pseudo-bulk, where you're averaging all cell types together. Also, for annotating the cell type, there are two methods being used. One, you can use an external biomarker, or you can use k-means clustering. What we need is a method that separates subject variability and cell variability, incorporates the cell type, and allows us to test for the various hypotheses I just mentioned. So incorporating cell type is important for detecting proteins whose expression relies on cell type. So that's what this graph is showing. It's showing a differential protein that is going up in the green cell type and down in the blue cell type. So it is differentially expressed, but only when you'll only detect it accounting for cell type. As soon as you average these cell types together, you obscure the difference. And I'll refer to this as the X pattern throughout this presentation. All right, so the data sets we used two. I simulated a data set, so we had known positives and known negatives, so we could track what the true positive and false positive rates were doing. And in this data set, I generated it to have two conditions, 10 healthy subjects, 20 diseased, 2 cell types, and 1,000 proteins per cell. So 980 of these proteins are our true negatives, so they're not changing between cell type. 10 proteins follow the X pattern, and 10 proteins are increasing in both cell types. And to show that the results we see with the simulated data remain true for a real data set, we use this biological data set from Sarah Parker's lab at Mount Cedars-Sinai that has two conditions, wild-type mice and mice with Marfan mouse aneurysm. We have 3 female mice and 3 male, but here I'll just be showing results for the male mice. 5,000 cells total and 5 cell types, but in this we only annotate the fibroblasts and the smooth muscle cells. All right, let's get into the results. And this first section, I'm just going to focus on how to actually incorporate this additional level of replication. So this is the main work that we did, is formulating these group comparison models. So on the right-hand side are models that capture what people are currently doing, and on the left-hand side is going to be our recommendations. So we characterized each model, we summarized the assumptions that each model relies on, and then we formulated the hypotheses. So these first 3 ignore the cell type. These next 3, they subset cell type to kind of account for, well, I know that if I average all cell types together, I'm going to be losing information, so it selects for one cell type. But then you're losing power because you're getting rid of, obviously, a lot of your data. And then our recommended model, it incorporates the cell type, but it does have to meet additional assumptions that your cell types will have equal variance. All right, so for the sake of time, I'm just going to go through 3 models, models 1, 2, and 7. So model 1 is when we use cells as biological replicates. So this is the data frame I showed earlier that shows all the features we need to incorporate. And this is what it looks like when we treat cells as biological replicates. So notice here we have 4 subjects and 16 cells. Here we're saying, okay, actually I have 16 subjects, and basically each subject is giving me 1 cell. So this not only gets rid of cell type information, but it also means that we're telling the model we have far more data than we actually do, which overfits the model and will inflate our false positive rate. So we can clearly see this in the simulated data. So on the right-hand side, I'm showing the true positives with this pink line showing the proteins that are increasing in both cell types, and this blue line showing the X pattern proteins. And if you just look at this, you might think, actually, this is a great model because I'm detecting a lot of my true positive proteins. But if you look over on the other side, this is our false positive detection rate. And you can see out of the 980 true negatives that we have, 250 of them are falsely being detected as changing. And I also generated data sets with increasing variability along this x-axis just to see what variability does to our results. And you can see that even when we increase it a little bit, we're at 50% false positive rate. So it doesn't matter that I detected 10 true positives. They're just kind of lost in the overwhelming amount of false positives that we found. So we see this we ran this on the mouse data set too, and we're finding more proteins than we expected to find, although we obviously don't know what here is a true positive and what is a false positive. Just given what we know about the model overfitting the data in the simulations, we suspect that a good chunk of these are false positives. All right, so the next model is pseudobulk. And you can see right away that we're treating subjects as the biological replicate, which is good, so it won't have that overfitting problem. But it is still averaging all the samples within a subject, so you're not going to get your cell type information incorporated, and you are combining your subject variability with your cell variability. So you can see right away, because we're not incorporating cell type, we're not detecting any of our X pattern proteins, and our sensitivity for the proteins that are increasing in both cell types is pretty low. And this is really seen in the mouse data set where we're not able to detect any differential proteins. And we do expect, we know there are differences between the disease mice and the wild type mice. We expect to see something. All right, so finally, I will present our recommended model, which incorporates all the features of single cell data. So it uses subjects as the biological replicate, it separates between subject variation and cell variation correctly. It incorporates the cell type, and it can support multiple hypotheses. So I talked about examples of these hypotheses earlier, but I'll just generalize the 3 types we can test. And the first one is we can compare conditions 1 and 2 for each cell type. So how is the green cell type changing and how is the blue cell type changing? We can take it a step further and compare the difference of differences. So how much more is the green cell type changing in comparison to the blue cell type? And finally, we can compare the differences between cell types. So how is green cell type different than blue cell type? These are the results of comparing conditions within each cell type using our recommended model. And you can see that now finally we are detecting these X pattern proteins, and that also our sensitivity for the increasing proteins is much higher. And these are the results on the mouse data set. So we did it for the smooth muscle cells and for the fibroblasts. And you can see in the smooth muscle cells, we didn't really find any differentially expressed proteins, whereas in the fibroblasts, we did. And this is a distinction that's impossible to make with bulk data. All right, so now that we have the model that we want to use, we're going to go on to talking about how to appropriately annotate your cell type. So there are 2 methods that people are using. One is you can use an external biomarker, which is where you have a protein that is known to be expressed in the cell type you're looking at. And so you use that marker, that protein, to mark the cell, and then you remove it from your data before going on to group comparison. And then the other way is you use all your proteins to cluster using k-means or something, and then you kind of use that marker to say, okay, I think this is my smooth muscle cells, and I think this is my fibroblasts. And then you take those same proteins you used to cluster to go to group comparison. And this leads you to a problem known as double dipping, where you are creating clusters and then using those same proteins you used to cluster to compare. So the analogy Olga likes to make is you tell tall people to go to one side of the room and short people to go to the other side of the room, and you say, are there differences between these 2 groups? And of course there are, because you design them to be different. So because you are designing these clusters to be different and then testing for differences, you are inflating your false positive rate. So again, we see this in the simulations. This top row is when we use a biomarker protein to annotate our cell type, and we get 100% accuracy. And this bottom row is when we use the double dipping problem. And even though we're finding some true positives, again, our false positive rate is so high that our true positives will just be lost in the overwhelming amount of false positives. So finally, we did this for the mouse data set. We did both methods of cellular annotation. And this side is showing when we use the external markers and then remove them. And then this is showing the k-means and then using all those again to cluster. And you can see that there's very little overlap between these 2 groups. And also given what we know, like the problems that come with double dipping and that we know it leads to an inflated false positive rate, we're fairly suspicious of the proteins that are found just by the k-means clustering method. All right, so I'll leave you with our recommendations. We recommend using a linear mix model that distinguishes cell types using external markers and not just averaging all your cell types together. We recommend you have a well-defined question, because as you can see, there are a lot of questions and a lot of different models you can use. And so if you just go and use all of them, you'll get into p-hacking territory where you just go and go until you find an answer that you like. Also, it's important to check your assumptions, such as equal variance. And you can just leverage any standard implementation of this, such as MSStats, which is what we did all of this work in. All right, I'd like to acknowledge everyone that was involved in this work, Olga Vitek and her lab, and everyone in Sam Payne's lab, and finally Sarah Parker and her lab for generating the data, and of course our sources of funding, the NIH and BYU Simmons Center for Cancer Research. I will take questions.
I would love to hear your perspective afterwards on metabolomics, but maybe more general. So how do you think data structure affects what you just talked about, missing data, technical noise? Are there additional considerations?
So for technical noise will be, because I talk about your subject variability and your cell variability. And in the model, your technical noise is like wrapped up into your cell variability. We don't have a way of distinguishing those two. And then all of our simulations were done just with like 100% data completeness, because I didn't want it to be like, oh, this is a missing data problem. This is a problem you would have and things to consider even if like you had 100% complete data. But yeah, I'm sure that missing data will give you trouble, So
Would you make these same recommendations for single cell RNA sequencing community, which seems to
Yeah.
me is the
no, yes, this is not specific to proteomics. All of this is whatever single cell thing you're doing, this is applicable to it. It doesn't matter what omics you're in.